Hello students, welcome to another lecture of pharmacology, and in today's lecture we will be discussing about pharmacodynamics. So in pharmacodynamics we basically study the mechanism of drug action. It is the study of drug effects. It starts with describing what the drugs do and goes on to explain how they do it. Thus, it attempts to elucidate the complete action-effect sequence and the dose-effect relationship. Now, there are certain principles of drug action. Drugs do not impart any new function to any system or any cell. They just alter the pace of ongoing activity. However, this alone can have profound medicinal as well as toxicological impact on a human cell. The basic types of drug actions can be broadly classified into stimulation, depression, irritation, replacement and cytotoxic action. Talking about stimulation, it refers to selective enhancement of the level of activity of a cell. Talking about depression, depression is a selective decrease in the activity of a specialized cells. Irritation connotes a non-selective, often nauseous effect and is particularly applied to less specialized cells. Replacement refers to the use of natural metabolites, hormones in deficiency state, and cytotoxic action mainly refers to the action on invading parasite or mainly cancer cells, attenuating them without significantly affecting the host cells. And this is mainly involved in curing various infections and various types of cancers. Now, coming to the mechanism of drug action, majority of drugs, they produce the effect by interacting with a discrete target biomolecule, which is usually a protein. Such mechanism confers selectivity of action to the drug. Functional proteins that are target of drug action can be grouped into four major categories. The first being your enzymes, second ion channel, third transporters, and fourth receptors. Now firstly, coming to your enzymes. So almost all biological reactions, they are carried out under catalytic influence of enzymes. Hence, enzymes are a very important target of drug action. In this image, you can see the enzyme has a binding site. When a substrate binds to the enzyme, product is produced. Therefore, an action is produced in the human body, and when an enzyme is attached with the inhibitor, inhibitor is a substance which inhibits the action of the enzyme. So, no product or non-functional product is produced. So, this is how drugs also work, by attaching to the specific enzyme. Now, in this image, you are able to see that when an agonist binds to a receptor, action is produced, and when an antagonist binds to a receptor, no drug action is produced. The definition of agonist and antagonist have already been discussed with you in the previous lectures. Now, the second type is your ion channel. In this particular image, what you can see, there are two molecules, one your blockers and the other your openers. Right? So when openers combined with your ion channel, the ion channel opens and ion can easily move from one area to the other across the ion channel basically. When blockers bind to the ion channel, it blocks the movement of ions. Now there are certain proteins which act as ion selective channels, as we studied in the image in the earlier slide. This makes them a common target of drug action. Drugs can affect ion channel, some of which actually are receptor because they are operated by a specific signal molecule either directly and are called as ligand gated channels, or G protein regulated channels. Drugs can also act on voltage operated and stretch sensitive channels by directly binding to the channel and affecting the ion movement through it. Right? So by two mechanisms, drug can act with the help of ion channels. Now the third type is your transporter. Now there are certain transporters in the cells which have structure in which substrate or inhibitor can attach. So when a substrate combined with a transporter, that substrate is transported from one area to the other, as you can see in this image. But when an inhibitor combines with a transporter, there is a variation in the shape of the transporter. Therefore, a substrate cannot combine with the transporter and no transfer occurs in this particular state. Now, there are several substances which are translocated across a membrane by binding to a specific transporter. Transporters are mainly your carriers, which carry a molecule from one area to the other, which either facilitate diffusion in the direction of concentration gradient, or they pump the ion against the concentration gradient, as we have seen in the image in our previous slide. Now, many drugs, they produce the action by directly interacting with the solute carrier class of transporters to inhibit the ongoing transport of the ion. Now, last type is your receptor. Now, there are certain receptors which are present in the human body. If an agonist combined with the receptor, the effect is further produced. Then there is a competitive antagonist and non-competitive antagonist, as you can see in this particular image. So, competitive antagonist basically combines to the same site where the agonist has to combine, and they both compete. Agonist and antagonist, in case of competitive antagonist, they both compete for the space. Whoever reaches the receptor first, they will produce the effect. So when an agonist will bind to the receptor, then the effect will be produced, and if competitive antagonist combines to the receptor, no effect will be produced. So talking about receptor is defined as a macromolecule or binding site which is located on the surface or inside the effector cell that serves to recognize the signal molecule or the drug and initiate the response to it. And there are certain terms which are used in describing the drug receptor interaction. One being your agonist, second being your inverse agonist, next your antagonist, partial agonist and ligand. Right? So talking about agonist is any agent which activates a receptor to produce the effect. Right? Inverse agonist is an agent which activates a receptor to produce an effect in the opposite direction to that of the agonist. Antagonist is any agent which prevents the action of an agonist on a receptor. Partial agonist is an agent which activates a receptor to produce submaximal effect but antagonizes the action of a full agonist. And ligand is basically to bind. So ligand is any molecule which attaches selectively to particular receptors or particular site, that is termed as ligand, and the term only indicates affinity or the ability to bind without regard to any functional changes. So ligand is basically a term used to define the affinity or the ability to bind. So thank you so much, dear students. This was all in today's lecture. If you have any queries, any doubt in today's lecture, please feel free to reach out to me.